Good morning, everybody. Thank you for being here. It's a real pleasure presenting an update about Acuamark to you. My name is Peperstraete. I'm co-founder and CEO. And we have, for the past years, worked on technology that we believe unlocks first-line blood-based early cancer detection. As it stands today, with the tools that we have, more than 60% of CRCs are detected at stage 3 or later, with survival of 50% or less. The compliance gap is 40%, so 40% of individuals are not undergoing screening. And we hope that a blood-based CRC screening test that is highly accurate, but marries high accuracy with high throughput, high margin, and universal platform access, which is fast and which is accurate, and does not exclude individuals at high risk. We hope that that will really help fill the compliance gap quickly. So we are currently out for a Series B investment. We've just started that. The market, of course, is large. Everybody, I think, knows that. We count about 175 million panels across South Korea, Japan, Europe, and the US on a biannual testing basis. We have verified our assays in clinical results. We have achieved best-in-class results. And our Series B uses really to further confirm these results and push them up to levels that we are comparable to market leaders. I move from about 300 samples we've tested and bring them to 3,000. We will be ready to start a PMA at points at the end of this use of proceeds, and we'll be M&A ready. So the platform, everybody is aware of qPCR, so it's an ultra-robust technology. It's the workhorse of the industry, but it's really limited to use in quite simple type of diseases, such as infectious diseases. And so we've worked to really optimize for the first time now qPCR to allow for broad panels of carefully selected ultra-low abundance methylation markers. So for the first time, it's now possible to easily and accessibly sample methylations with just qPCR. Of course, we have improved the assays such that we get to better accuracies, but we have really worked hard on maintaining that high scalability, that broad access, and the low cost of goods of qPCR combined with machine learning. The first products are really focused on CRC screening and CRC recurrence. The data I will show you are still with based on just qPCR combined with predictive models. The assays are CIMA automated, they are stable. And with the Series B, we will transfer them to a development lab. So to give you a peek on the results, so in the past 6 months, we've worked on the assays and finessed them slightly to further economize on primer usage, so that lowers the cost of goods even further. And so I'll show you, we've applied those new methods on a 132 case control set, the latest set. Before that, we had a 216 case control set, which I'll show you results on. And the confidence scoring models that we have allow us to identify borderline cases to advise retesting in 3 months. So, so here you see some of the results that we've presented before. You see several of the models have sensitivities in the 92 range, the best ones, with specificities ranging between 95 and 97. You break that down to stage 1 through 4. Again, this is CRC, and see several of the models having sort of atypical detection rates for stage 1 and stage 2. Several of them detected all of the stage 1s and the stage 2s. And on average, throughout the various models, we see that the performance remains very strong, which tells us that the biological inputs are there. So here are some of the confidence scoring that we did on these 216. You see that we have pretty much detected all of the stage 1s, the stage 2s, the stage 3s, stage 4s. We had a couple of missed calls on the healthy side. On that for you, it's the left side. But we believe that perhaps amongst that pool of healthies, there might have been a couple of adenomas, perhaps, or perhaps cancer cases that we couldn't yet detect because the controls were not colonoscopy confirmed yet. And here you see the latest set. So this is an update. We're very proud of these results. This is with the newest version of the assays, and you see that pretty much Again, we detected all of the cases quite accurately. There was a couple of borderline cases you see that sort of go within the 0.4 to 0.6 range. These individuals, we would know that these individuals would be borderline cases, and we would advise them to come back in 3 months. Again, in the healthy pool, there was one that came out with very high scoring, which we couldn't yet confirm whether that was truly a healthy individual. Here you see a representation of the 40 markers that we're using, 38 of which are, we think, really knocking it out of the park. You see that there's a consistently higher scoring amongst the CRC cases relative to the healthy cases. You can compare that to normal qPCR panels of like 10 markers or so. So this is what we've done. We're currently working on 80 plexes. If you compare the high sensitivities of 92, 97, the low cost of goods, the high throughput, fast turnaround type of approach with other approaches, you see on the next-gen sequencing side, there's really a ceiling in terms of sensitivity, 83% sensitivity, 79% sensitivity, at 90% specificity and 91% specificity for some of the other approaches that have been developed or are in development. We see on the stool side, Cologuard is reaching very high levels of sensitivity and specificity, which we really applaud. We do think that with a blood test that comes within striking distance of these accuracies, the compliance gap can be filled much faster. This technology is, of course, designed to seamlessly fit in a medical workflow. So eventually, this will just be a kit that's sent out to every lab that has a qPCR instrumentation workflow. And we believe that for administrators, this is, of course, easily integrated into the medical workflows, but it also focuses their resources on where the need really lies. For patients, it's easily accessible, it's highly accurate, consistent peace of mind. You can easily imagine this being used in first line next to your lipids, HbA1cs where it's relevant, etc. And for payers, it really dramatically lowers risk and exposure. So for CRC screening, we envision an FDA path, a PMA study, which we'll hope to start in the end of 2027, beginning of 2020. End of 2026, beginning of 2027, and the CRC recurrence product will be a 510K. Large market, so 175 million panels. This is well published and studied. And we're focusing on CRC screening and CRC recurrence first, but then, of course, this platform allows us to go to other cancers next. Again, we believe that this highly accurate approach married with the scalability lifts the limitations to bring accurate first-line cancer screening into the mainstream. Very proud of our team. We have focused it in a functional way around the clinical. We're doing a large collection currently, 3,300 CRC cases, and then, of course, on the laboratory team, which contains my co-founder, Francis Barani, who is the inventor of ligase chain reaction, ligase detection reaction, etc. This market has been very dynamic, and so we've seen Natera overtake Exact Sciences, and we believe that our current plan will allow us to get a good profiling against these approaches. There's also been a very good reaction from the public markets in terms of multiples, in spite of net incomes that are sort of ballooning as well. And then we see in the pre-revenue sort of spheres, we see some past transactions. Aims for the Series B will be to get the CRC clinical validation to 3,000, be PMA star ready and M&A ready. And in summary, the approach really embodies a high blood-based accuracy, but at the same time, a consistently strong sensitivity for early cancer detection, which is where this approach starts separating itself from other approaches. But at the same time, keeping also low cost of goods alive and make this easy to scale. We believe this will really result in high compliance, and then importantly, patients at risk are not excluded. Thank you so much for your attention.